last speaker, I had to hurry up. I didn't pick her up, so that's why she's on time. So yeah, it's my pleasure to introduce Dr. Kalpana Katti. She's a University Distinguished Professor of North Dakota State University, and she leads there the NDSU Center for Engineered Cancer Test Beds, and also NSF EBSCOR Center for Cellular Biointerfaces in Science and Engineering. She has a PhD in Material Science and Engineering from Washington, Seattle, University of Washington, Seattle. She is an NSF Career Awardee, has multiple, awards, but I'll just read a few. She is the elected fellow of the American Institute for Medical and Biological Engineering, recipient of Phi Kappa Phi lecturership. She'll be talking about her primary research area in tissue engineering, cancer scaffolds, biomimetics, and her work is also in multi-scale modeling, experimental studies on biological systems, natural systems, as bones and shells. Her group has made important discovery, such as third-tier hierarchy of collagen and the interlock mechanisms in shells. Obviously, she has published hundreds of papers, given many invited lectures, keynote, and preliminary talks. So we are very excited. I have listened to her lectures before. It's an amazing talk, so I'm very excited to have her here. Welcome to Miami, and thank you.
Thank you, Dr. Prasad, for the very kind introduction. I think my mom would be proud. And it's a pleasure to be here. I come from the frozen tundra, so it's really wonderful. I was just outside. I came a little early. I was just walking around. It's nice to be able to breathe and not be so. You have no idea what I'm talking about. Compared to what minus, 40 degrees with wind chill. So it felt really nice. I'm going to talk about cancer test beds to look at this very important medical problem from the point of view of engineering and our engineering approach towards this serious problem. And I'm going to talk, I'm an experimentalist, I'm going to talk extensively about experiments, but this work that I'm presenting is guided by a lot of simulations work, modeling, and so I will kind of touch on it a little bit, and that part is done by my collaborator Dinesh. And of course, it's the work of a large number of graduate students. So just want to show you where I traveled from. So that's a world map, that's North Dakota, and it's right there by the Minnesota border, okay? And it's actually pretty nice most of the year. It looks like this. It looks like just like this right now, but it's monochromatic. So coming to cancer statistics, we're looking at about over 3.3 million people that are diagnosed with just these two cancers, breast cancer and prostate cancer. And out of this, 1 million people die each year. So 1 million deaths are credited to prostate cancer and breast cancer alone. And in most of these cases, the deaths are associated to a process called metastasis, where the cancer transports to a distant location in the body. And when that happens, the cancer is incurable. So at that point, there is no cure for cancer. So as of today, there is no cure for metastasis. We're looking at a very severe problem. About 1 in 8 women and 1 in 7 men are likely to get prostate cancer or breast cancer in their lifetime. Okay, so that's a little cartoon that shows where different types of cancers transport to in the body. So breast cancer and prostate cancer have the propensity to transport to bone. At this point, the anti-cancer drugs are ineffective. Animal models are of no use, because by the time the cancer goes to bone, before that the animal is dead. It's very difficult to get samples from patients. At this point, they're in a very late stage of cancer and often in a hospice, so it is hard to get samples. So that necessitates the need for a sort of an engineering test bed. So what we want to design is an engineering test bed that is to create in a laboratory the situation of bone metastasis of breast cancer and prostate cancer. A little schematic showing sort of the process, and up here at the top is a photo of the bones of a person when the prostate cancer or breast cancer arise at the bone site. And very porous, they break easily, skeletal failures. I could show you more photos, but they're just too gruesome. At this point, the surgeons or the medical professionals are basically treating the bones. So they're putting like pins and plates and to sort of stabilize the bone because there is no cure. So basically the cancer starts at a primary site over here, it grows, and this is where all the early detection and all the drugs are competent at killing the cancer. But in a few patients, a few of the cells lose adhesion, get into the bloodstream, travel to a remote site, and that happens to be bone in this case, and then colonize there. Okay, and not only there's a tumor there that has to be handled, but they actually affect the bone. So the focus of our work has been to look at what happens to the bone really. There are very specific gene markers that are characteristic of this first stage, which is the primary site, and then there are very specific gene markers that are characteristic of the secondary site. So by just analyzing the tumor, you can determine what site, what condition it is in. This latter site is called as MET or mesenchymal to epithelial transition. Okay, so I'm going to say a lot of things, a lot of different ways in which we are tackling this. It's a very long story, but certainly it's a very important one given that 1 million people die each year. So what I'm going to talk about is basically design of first the bone mimetic scaffold. So first we make the scaffold, we mimic the bone using traditional tissue engineering approaches. Then we create the remodeling. There's a particular type of bone to which cancer metastasizes to, so we create a remodeling bone. Then we make what is called as in vitro models or test beds, where on this remodeling bone we create tumors by using either commercial cancer cells, human cancer cells, or patient-derived cancer cells. So we create the tumors in the lab. And then now we don't have to poke a patient. We can get these samples out of the tumor literally on a daily basis. So we can use this to design or develop new biomarkers of metastasis. Because as of today, we don't have any biomarkers for metastasis. We also want to make this system as real or human as possible. For this, we use our specially designed bioreactors that sort of simulate blood flow and shear forces exerted by the blood flow. And of course, all of this is guided by simulations, both the design of the materials, molecular phenomena like cellular adhesion, things like that. So that's kind of all the different chapters of the story that I'm going to try and go very, very briefly. Certainly, tissue engineering needs no introduction to this group. I'm sure you've seen these diagrams before. The advantage is basically there's no rejection. The tissue is made using patient's own cells. So we have basically 3 ingredients of tissue engineering: the scaffold or the mesh that degrades the cells, and then the growth factors. And of course, we use bioreactors to simulate blood flow. And we've worked extensively on design of the scaffold system with a variety of different materials, and also manufacturing techniques. So from bio-based polymers to synthetic polymers and such. To give you a little background on the special type of material that we use to make scaffolds, it is a polymer clay nanocomposite. So we put nano clays into polymers, and that improves the mechanical property of the polymer several hundredfold. Okay, so several 100%. So several fold, I should say. So, and the reason for that is really PCNs, or polymer clay nanocomposites, were first proposed by the Toyota Motor Company for timing belts of their cars. And they found that if you add a small amount of nanoclay to a polymer, you can immensely improve the property as compared to if you use the same amount of clay, but it's not nano, okay? But why that happens really came about through extensive work about 3 of my graduate students, and we came up with this fundamental theory called as the altered phase theory, and it basically describes why nanoclays improve the mechanical properties of the polymers. They actually alter the polymer to a great distance away from the nanoclay itself. So if you see this sort of a sunny side up egg model looking like schematic over here, where the yolks are the clay particles, and darker blue is the region where the polymer is influenced. So larger volume of the polymer is influenced. Of course, this work came about through extensive molecular dynamic simulations, and that's kind of what is shown over here in this diagram. Those numbers are interaction energies in kilocalories per mole. So through all this, we were able to describe that. The problem is that to make a polymer clay nanocomposite, these clays are layered silicate structures. You have to kind of send in small molecules called modifiers, and then they actually pull in the polymer. So it's a three-phase system: the nanoclay, the modifier, and then the polymer. A lot of the modifiers used are for PCNs are actually toxic, so we can't use them for a lot of applications that are in biomedical. So we decided to use, again, this was also guided by simulations. We started to use amino acids and to see if they intercalate. Well, it turns out they do intercalate. That's what our simulation showed. But the simulations also showed, and so did experiments, that the modifier needs to be a little bit bigger molecule to influence the properties. But amino acids are really tiny molecules. So what we did is we started to use analogs of amino acids that are called unnatural amino acids that are synthetically made, and they're just longer with more carbon chains. And that's what we used here. We picked 3 that are shown by the pharmaceutical industry to work very well. So these also show, and of course we can improve mechanical properties. At the end, the cells have to like it. So these are some tests done with osteoblasts. So we decided to go ahead and use these unnatural amino acids and modify the clay. Now, interestingly, in order to make this osteogenic or bone growing, remember we're trying to grow bone here first, we have to throw in some hydroxyapatite. So it becomes a really complex system. Clay, amino acid, polymer, hydroxyapatite, and that's not very biomimetic. So in nature, in biology, bone is grown, or hydroxyapatite is grown, sort of through the use of non-collagenous proteins on site. So the idea was, we already have these amino acids, and we could use these amino acids inside the clays to mineralize hydroxyapatite. That was just an idea. And some extensive work by my grad student Avinash, it turns out it worked. We were able to, what you're looking at here, these spherical shapes, these are basically hydroxyapatite that's grown inside clay galleries, and this has a morphology very similar to biomimetic hydroxyapatite. So now we have a single phase system: clay, amino acid, hydroxyapatite, and then stick it in the polymer, and that becomes our scaffold. And so that's more biomimetic. But another thing is, how does biology grow hydroxyapatite in the biological process? So these individuals got the Nobel Prize in 2013 for describing how this happens. Is in the cell throughout little packets called vesicles into the extracellular matrix, and these vesicles carry the seed of the mineral, and that's how mineralization begins in biology. We wanted to see if we are seeing this process in our scaffold systems. So we did see these particulates. These look like the shape of the vesicles that are seen in biology, but again, through a very clever experiment by Avinash, basically scraped out those vesicles, put it on a TEM grid, and indeed it has hydroxyapatite. So it has hydroxyapatite. So what we've actually made is biologically, chemically, in every way, mechanically, very similar to bone. It's osteogenic. And not only that, if you look at the calcium to phosphorus ratios, it has a sort of characteristics of a remodeling bone. So calcium to phosphorus ratio for remodeling bone is lower. So this is exactly the type of bone to which the cancer cells metastasize to. So of course we are using this for regenerative medicine and looking to license a patent, and those kinds of efforts are going on. But coming back to cancer, now that we made bone on these scaffolds, it is very much bone-like in every way, and to this now we insert the cancer cells. And when we do that, we were able to generate these tumors at the bone site. So that's our test bed. Here's a picture of a prostate cancer tumor at the bone site. This is made in the lab. If this were to be in a patient, they're looking at a very short life, okay? But we are looking at this creation of this. This is another photoconfocal image of prostate cancer tumor at the bone site. And again, we wanted to investigate from the tumor what it is, what condition it is in, and analyzing the genetic makeup, gene expressions of the tumor. We find that it is in this late stage metastasis, or MET, or mesenchymal to epithelial transition stage. So this was the first ever MET stage in vitro test bed of prostate cancer, and we were able to do the same with breast cancer. So here what you're looking at is a breast cancer test bed with these images on the right are breast cancer tumors, and you can see we can extract these tumors on a daily basis now. Okay, and we can watch for morphologies, and we can see how they grow, and all of these that is not possible from patient samples. So over here you're looking at, for an example, two kinds of prostate cancer cells. PCA, the low or less metastatic. PC3 is the highly metastatic one. You can see at the bone site, they make very different kinds of tumors morphologically. Even just looking at these pictures, you can see one is kind of a dense type tumor, and the other is sort of loose, or sort of more loose aggregates. So very different characteristics. That was prostate cancer. Same thing with breast cancer. The top row is MCF7, low metastatic breast cancer, and the row at the bottom is the triple negatives, the really bad breast cancer. And you're looking at the bone site. And again, you can see very different morphologies of the two. Okay, so what is happening to the cancer cells when they arrive at bone? One of the reasons we cannot cure the cancer is because when it transports to the bone site, is a completely different animal. It is genetically very different. We really don't understand that tumor. So one of the things we wanted to do is to understand this tumor. One of the characteristics is the migratory nature. So we designed a simple sort of transwell assay method to measure how much more migratory it has become. So triple negative tends to be more migratory than the MCF7, which is a low metastatic. So in a sense, we knew that the cells themselves are more migratory than the less metastatic cancer. But if you look at what is happening to the bone site, we're seeing for both kinds of breast cancer, the presence of the bone site or arrival to the bone site makes the cells even more migratory. So arriving at the bone site is actually influencing this. So breast cancer types influence the bone differently. So not just that the cancer cells are altered, but we also were looking at what happens when they arrive at the bone site. And in order to do that, we looked at the fundamental osteogenesis pathway and see that when the cancer cells arrive, and whatever it is that they secrete, are they influencing the bone? So remember, the patient's bones are breaking. So a lot of effort goes in killing the cancer, but really the problem is the bone. So what is the effect on osteogenesis of the arrival of the cancer cell? So we sought to figure this out. One of the important sort of pathways is this Wnt pathway, and what we found is there's an upregulation in the Wnt-related factors in the bone metastatic cancer. So if you look at all of these four plots here, the dark bar is the healthy bone. Okay, that's how we want it to be. And the other two are bones that are around the cancer, two different types of cancers. So two different types of cancers, and this is the healthy bone. And you can see in all cases, this one type is higher and this type is lower. So arrival of the cancer cells has influenced osteogenesis very significantly, at least from the point of view of event-related factors. Now these breast cancer cells secrete some very special factors called DKK1 and ET1. And very simply, I want to say is one of them acts like an enabler of this process, and the other acts as a blocker. So what is happening is when these cancer cells arrive at the bone site, one type of breast cancer is putting in more blockers, and when that happens, we have reduced bone formation. And in the other case, we have excessive bone formation. And that's exactly what we saw experimentally, also clinically it's observed. So this is kind of how the mechanism by which the cancer cells are influencing the formation of the bone. So fundamentally how the bone formation is happening is not normal. So what we want it to be is like the healthy bone, but it's not, okay? So now that was breast cancer. We look for the same things with prostate cancer test bed. What you're seeing here is the bone near the tumor. So we're looking at the bone near the tumor. I don't know if you can see a lot more red in this case compared to here. So this is how we want it to look like. In one case you have more bone formation, in the other you have less bone formation. So this is just staining the mineral. Now if you stain the collagen, the other component of the bone, bone has mineral and collagen. So again, you have excessive collagen here as compared to healthy bone, and you have reduced collagen here as compared to the healthy bone. And again, we are able to monitor this on a daily basis, basically. So, interestingly, you have excessive bone formation or reduced bone formation. So both are bad. So that's what was observed. But and again, all of this data just together. So both the mineral and the collagen. So you have either excessive bone or reduced bone, kind of like what we saw for breast cancer. So that's why the bones break. The other interesting thing that we are pursuing to look at is bone is made up of these mineralized fibrils. I don't know if you're familiar with bone is made up of collagen, and then it has a very interesting hierarchical structure, and that has a great deal of detail and perfection. If you look at a healthy bone, this is what it looks like. There's like a banding pattern that's 67 nanometers exactly in healthy bone, exactly. In fact, when you have severe genetic bone diseases, that's when this pattern is disrupted. So your two arms may not be exactly the same length, but at the molecular level, it is absolutely accurate, this banding pattern, okay, 67 nanometers. Now if you look at the fibrils near the tumor, so these are the mineralized fibrils near the tumor, nowhere like this perfect healthy bone. So we are actually this is a very scary result. It almost suggests some sort of, the treatment for this is not just replacing bone, because it's genetically altered, okay? So, so we're pursuing this a bit more. Now that we have this ability to take the tumors out on a daily basis, what do we do with it? So we have literally on a daily basis, we can extract the tumor in a Petri dish and then try and find some markers. One of the methods we wanted to use was using IR, and the reason for that is IR can be done remotely. It can be done without a biopsy through the use of fiber optics, so it's very commonly used. So of course we don't need to use fiber optics, we can actually take the tumor out, but essentially the idea is we can get this from a patient without a biopsy, okay? So some very detailed analysis was done. Very simply, we, on a daily basis, on 2 kinds of breast cancer, we were able to take FTIR data. This is the whole spectrum, 5-day, 10-day, 15-day, 20 days, like that, and analyzing this. So we were looking at 3 very specific regions: the lipid region, amide region, and nucleic acid region. With a great deal of detail. So of course, there are not great deal of differences. So these are very minute differences. We wanted to see if we could find any statistically significant differences in these regions. So we did a very detailed principal component analysis And we're seeing that these three regions, and specifically for these regions and the ratios, so we are looking at the amide ratio, RNA, DNA ratio, and the lipid content, and see if there are differences in this as the metastasis progresses. So we're using FTIR to give a measure of progression of metastasis. FTIR has been used before to tell the difference between cancer cells and healthy cells. But this was the first time looking at not just metastatic cells, but at metastasis. And basically the principal component analysis showed that we're able to show these differences. So it has, of course, all of this analysis, little tiny programs written, all connected, and all this. For this to be an actual product, it needs to be packaged in a different manner, kind of like a software that is easily usable. So that's kind of where we're at. The other things we're doing is we're shipping our tumors to different labs. Here we made those tumors on, again, 5-day, 10-day, 15-day, 20-day tumors, ship them FedEx overnight from Fargo, North Dakota to Boston. So it was shipped late in the evening, and my collaborator was waiting there at 8 a.m. At the MIT Raman lab, one of the most sophisticated Raman imaging facilities in the world. And collected the data, sent the data back. This is a great deal of AI and computational analysis involved. I won't get into, but bottom line is we were able to show sort of a label-free method. So there's no staining. This is very similar to what you do with confocal, but for confocal you need to stain the sample. So this needs no staining. So this is a label-free evaluation of extent of metastasis. So this is kind of where the world is going, label-free, remote, without using any biopsy samples. So this just came out last year with my collaborators. The other idea, some of the most interesting things I feel that we're working on is this field called mechanobiology, and I think the genealogy of that is right in the realm of Dr. Prasad's maybe a long time ago graduate work. I believe her former advisor proposed this, I think about 30 years ago at MIT, that cell health or tissue health has something to do with mechanics, or rather mechanics has something to do with the tissue health. So healthy cells versus disease cells, and they showed this for malaria. And since then it has been extensively evaluated using a variety of different evaluation methods of mechanics to use mechanics as a measure of disease in a cell. So it was known that cancer cells are actually softer than healthy cells. People have already shown that. So we wanted to see if this progression of metastasis can be described by mechanics. And our purpose was, it says biomarker, but really not to take the tumor out and measure its mechanical properties. Oh, you're gonna have cancer, like that just doesn't sound right. The purpose of it is to build a truly in silico or a computational model of the progression of cancer so we can give a predictive capability. So the purpose is more than just mentioning how the cancer is progressing, but to be able to predict it. Okay, so that's been our goal. So we used, it's been very common in literature to use AFM. So you use an AFM tip and the approach and retraction, but that doesn't engage the whole tumor cell. It only engages the surface of the cell. So what we tried is this sort of indentation method where we built our own system, putting in simple engineering actually. So kind of put in a heater and then some fluid, some chamber to hold the fluid. So underneath the isotron indenter. So we built that system, and then and why would we want to do that? Well, why did we think that metastasis is going to change the mechanics of the cell? When we looked at photographs of the cancer cells that were undergoing metastasis, so the progression of metastasis, we found that the inner workings of the cells have changed. And by inner workings, I mean the parts that are responsible for mechanical properties of the cells. There are many things in biology that different parts of the cell, right? But as mechanicians, we were looking at what are the parts of the cell that contribute to its mechanical properties. And they are very specific parts of the cytoskeleton that are now colored in here, the reds and the greens, okay? So these are the two parts of the cell that are most responsible for its mechanical properties. And our images show that these are changing as metastasis progresses. That means overall mechanical property of the cell must be changing. Okay, so that's why we went about to do this. So here's another photo where I think it's a very beautiful image, right? I would love to have a dress made of this, but they made this into a fabric, but it's a prostate cancer dress then. Who wants to wear that? Okay, so we did measure the mechanical properties. So this is some indentation data. I just want to point out, if you look at the top over there, we see that the tuberoid cells become softer with the progression of bone metastasis. So going from gray to pink to teal, you see that they're getting softer. So elastic modulus is decreasing. So tuberoid cells become softer. We're getting an accurate measure of the modulus. I just want to point out, this took a lot of work to get this published, by the way. And the reason for that is, look at the numbers. We're saying that the elastic modulus of the cancer cell is in the MPA region. If you look in literature, they're always showing KPA. So of course nobody believed it. Look, I think the response to Revere's comments in this particular one was like 18 pages, as long as the paper. And there was a lot of computational analysis done, a lot of calculations done to show that the cell is made up of a cytoskeleton. 27% of the cell is made up of a cytoskeleton which has gigapascal modulus. So it's kind of expected that it will be megapascals. So finally got published. I just want to show, this is what I've been talking about, the cytoskeleton. So the top row is a single cell, and these are the cells in the tumor. Notice these two things, the red and the green stuff, which is the actin and the tubulins. Notice the red is not only reduced in the cell, but it's also moved to the edges. So very significant changes have happened when the tumor is formed at the bone metastasis. On the other hand, this green stuff, not really big change. So the green is not really participating so much in the progression of metastasis, but the F-actin is. So we also looked at the genes associated with the F-actin. Seems like F-actin is such a player in metastasis. So, and just as our mechanics data showed, all the genes related to F-actin are downregulated. So no wonder, as the cancer is progressing, the F-actin is reducing per cell, and then we also have lowering of the mechanical properties of the modulus of the material. We also did some dynamic experiments. These are done on breast cancer for two different kinds of breast cancer. And notice in this one, this is the low metastatic one. In the low metastatic one, we see that's the top row. You see that there large tumors are formed, and you have a reduction in, notice the red color is reducing, right? So, and that agrees with the amount it is reducing, agrees with the lowering of the mechanical property. On the other hand, the triple negative really doesn't show any changes to the F-actin, and also its mechanical property doesn't change. So this metastasis cell is actually more resistant at the bone site. No wonder we can't cure the highly metastatic one. Okay, so basically the cytoskeletal softening that is arising primarily from the F-actin, it is what is causing the change in mechanical property. So the F-actins went to the edges of the cell, right? But they're not hanging by themselves. What sticks out from the F-actins are the molecules that connect the cell and cells. The more they go to the edges, the more they form the entanglements between cells. And these can be measured using the cadherins, and again, we do see, when we measure the cadherins, we see a direct correlation. So the F-actin goes to the edges of the cell, makes the cell more deformable, and then it's able to connect to other cells and increase adhesion. So we were able to understand this both for breast cancer and prostate cancer simultaneously. And of course, we wanted to just kind of verify, why is it that drugs don't work? Drugs are not working. None of the drugs we have are really effective at even killing the cancer at the bone site. Well, the bone metastasis enhances, so the presence of the bone enhances the multi-drug resistant genes. We actually measured this. We saw that no wonder it can't anymore fight the drug can no longer fight the cancer because the cancer cells are completely resistant. All this is using patient-derived cells. So, using commercial cell lines. I showed PC3, PC8, MCF7. These are commercially obtained cell lines, but we also have a collaboration with our local hospital. The Sanford system is the largest hospital system in the Midwest. And we also had a collaboration with Brigham Brigham Hospital at Harvard. And for 2.5 years, I mean, we still have that collaboration, but apparently it's easier to get samples from Sanford locally than from Boston. And the reason is every patient going in for surgery at the hospital at Harvard is their sample is spoken for. So it's really hard to get any tissue samples. So we weren't able to for 2.5 years, but we've been able to get about 8 patient tissue samples that we then grew. We made a cell line out of it and then we grew it on our scaffolds, just like I showed for the commercial cell line. So these are patient-derived cancer cells. They also form tumors. The top row is the ER positive, this is the triple negative, kind of like the MCF7 and MDA231. So sort of comparing, does the patient-derived behave just like the commercial cell line? And the answer was yes, and of course we kind of expected that, but the medical community doesn't have much respect when you use commercial cell lines. They want you to show your results with patient tissues, and that's what this is. So it agreed very well for the type of cancer. It agreed very well with the commercial cell lines. So we saw MET, we saw all of those. That just came out actually last week. So the student had 2 fabulous papers in the same week. It was too happy. And the things we saw with DKK1, ET1, one's a blocker, one's a enabler, all of those results are consistently seen with patient-derived also. So we saw the same results. So our test bed is very accurately able to represent creation of tumors at the bone site using both commercial cell lines and the patient-derived cell lines. The last thing is the interstitial flow, blood flow. What is a blood flow? Is it causing cancer? Is it preventing cancer? Blood flow is very important, right? So it's a healer, it's also an enabler of some things. Well, metastasis wouldn't even happen if we didn't have blood flow, right? But we wanted to investigate what is the role of the blood flow, both in the metastasis and also in actual application of shear forces to enable adhesion of the cancer cell to bone. Because it's just flowing about. Why does it want to go and attach to bone and colonize? We wanted to look at that. So for this, we designed a bioreactor. So this is a perfusion bioreactor you can see over here, easily scalable. For this 4-channel pump, we can easily scale it up quite easily. And it's able to duplicate these 2 important things: carry nutrients, remove waste from the cells, and also induce appropriate physiologically relevant shear stresses. So we needed to know what is physiologically relevant. What does blood flow, what kind of stresses it causes? So we looked at that. So again, some more images of the design of this. And it's very scalable. It can be, it is placed inside an incubator. But like I said, it's very easily scalable. Okay, so we did dynamic versus static. So we do dual experiments that are static. That's like normal. You take the media out every 2 days, kind of like a standard experiment. And the other is where you have this media flow. And we also take the scaffolds. In order to do a fluid dynamic study, we need to know the microstructure of the scaffold. So that was obtained using micro CT images. So just for the growth of bone, so we wanted to start from the beginning, because we first start to make the bone. So does the flow affect the bone growth? So the mesenchymal stem cells, that is MSCs, they generally show higher proliferation. So we see higher or increased proliferation because of the flow rates. Okay, so you can see over here, again, more cells are alive, so you see proliferation means more cells. And they look like some more pictures if you see. The one on the left is static and dynamic. This is the direction of the flow with the white arrows, and you can see so not only they are more they are more in numbers, but they also have a directionality. So these were some simple sort of observations. Now, what is happening to their differentiation capability? We see initially, so this is 10 day, 23 days, okay? So what we're seeing is for two of these important markers, ALP and RUNX2, we see elevated expressions over 10 days and then reduced. And then if you look at OCN, it is less and then increased. And basically what that means is that you have sort of early stage osteogenic genes, RUNX2 and ALP2 are, ALP are increasing and then decreasing. So you basically have more mature cells happening. So their state is in life cycle is different. So these are hallmarks of cell maturation, because this is the point at which at 23 days is when we put the cancer. So we wanted to see how we have influenced the bone even before the cancer arrives, because that is important from a biological standpoint. Okay, and some more detail about the cell culture analysis, cell cycle analysis. So if you look at the cell cycle, it goes through a first phase, which is non-dividing, then it causes division of the DNA, then it has division of the cells. So we can measure this out, and again, it's also agreeing that basically we are seeing that the dynamically cultured cells, that is in the bioreactor, have more mature cells. They have more mature cells due to the high differentiation rates of the mesenchymal stem cells. So that's a good thing, because that is where the cancer wants to go to. So it is interesting, means flow has something to do with it. That which is important for life is also causing cancer, basically. So again, same kind of a thing using other methods of imaging, confocal imaging and all that. So what is all this coming from, basically? We have seen increased MET markers in the dynamic. So you have seen more of this MET stage cancer happening in dynamic. You're also seeing altered cancer morphology. So there's like a tug of war happening between the E-cadrins, these are the ones that connect the cells together, the E-cadrins and the fat proteins. So you have this sort of tug of war happening, and that causes, so a lot of mechanics happening to alter the morphologies of the cell, because we do see changes to the morphology between static and dynamic. So these simultaneous pulling forces by adherins and integrins. Okay, now integrins come into play. So things that connect the cells are cadherins, and then the cells, when they attach to the bone, that is integrins. So there is that too happening. So of course the PC3 or the cancer cells become more migratory. We saw this again with our bioreactor experiments, just like we had seen with the static. This is a brand new one. We just filed a provisional patent for. This is a new horizontal bioreactor. This is a simple design or part of it. But what I wanted to mention is that we're able to duplicate exactly the physiological condition inside the scaffold. And this is why we needed to have the microstructure of the scaffold to be able to. Before I say too many, I don't want to go into too much details of this, but really what this bioreactor enables is something very unique. And that is that now we're not putting the cancer cell on the bone. We're letting the cancer cells go through a port. And if they like the bone, they'll go and attach to it. That is the natural state. And that's where our experiments are going on right now to do this. So this is even more natural. It's a more realistic test bed. So again, same things, migration of PC3s, lots of detail about these are some markers that tell how migratory the cancer cells are, and we are able to measure that. So bottom line is, when this cancer arrives, why does it attach to bone? It is attaching to bone through these molecules called integrins, and the integrin levels are increased under dynamic conditions, both in the presence or absence of. So flow rate is really causing, and is one of the reasons why the cancer cell attaches to bone. On the other hand, in the presence of bone under dynamic conditions, these integrin also contribute to increased levels of the migratory nature. So it's like a double whammy, basically. So you have more chance due to the flow, and you also have the presence of the bone itself, the characteristics of the bone that the cancer cell wants to adhere to. So both of these are playing a role, basically. So why, if you recall that actin is responsible for you recall that I showed this picture, that actin is really important for the mechanics of these metastasized cells. So this actin, we did a lot of analysis to model this actin itself. So this is where I said I'm not talking, this is surely a story for another day, but we're also modeling we have modeled the actin structure. It's like a collagen structure, which is a triple helix. This is a double helix, except that unlike collagen, which is like two very strong ropes, three strong ropes coiled around each other, these are like blobs. Each rope is made up of blobs. Those blobs are called as G-actins. And it's the coming together and breaking apart, so like this polymerization, depolymerization of this actin that causes this. And so that's what is happening internally in the cell, and a diagram that shows that. So why do they come together and break apart? There's this other guy called coughlin, and coughlin attaches and they come together or they fall apart. So it's important to model this with the coughlin. So coughlin is also present in this cell. So all these processes are starting internally, but they affect what is happening outside of the cell to decide whether they will attach to another cell through the cadrin, or with the integrins attached to another, to the substrate. A lot going on here, but I just want to say the coughlin very simply, the student, Sharadu did this, is a mechanical engineer. He comes up with very interesting mechanical analogs. So coughlin attaches to the attaches to this actin, and literally you've seen how to open a beer bottle, Budweiser bottle, like this, 30 degrees, right? And that's exactly what happens. It loosens it, it opens it, and that's how the polymerization, depolymerization starts. It's a very simple mechanical effect that we saw. Okay, so this is a lot again. The last thing is, so this is happening inside the cell, but the cell has to now adhere to the cancer. This cancer cell has to adhere to the bone, and like I said, that is happening to the integrins, and we have also modeled that. This is done by Hanmant. There is something called as this kind of lies by itself, this integrin that is sitting on the top of the cell, but something has to come and wake it up or activate it, and once it does that, that's when it latches on. So we are able to model that. We're actually looking at the clay surface or the bone surface. Why clay? Because our scaffold has clay, and bone because that's what we are making. Turns out that is actually influencing this activation. And I mentioned clay because we're also thinking therapeutics very fundamentally. We can use that to activate or deactivate the integrin. That will decide whether metastasis happens or not. Without this activation, you won't have metastasis. Anyway, this is all molecular dynamics simulations. We're also using the bone. This is kind of really cool little thing. Our university is trying to license this. This came about because of the Lego store. How many of you have been to the Mall of America? They have a giant Lego store there. We were there, and that was just the inspiration for it. These bone defects take a long time to grow, and that surgeons told us it's not realistic in an injury. So they said, why don't you speed it up? And so we came up with this idea of Lego blocks, a lot of finite element analysis to find what the connections are. But basically this patent that is showing what we did is we use Lego blocks, and we also use something called as bone morphogenic proteins. So we coated them. All I'm trying to say is we are able to initiate bone growth in as fast as 3 days, as opposed to like a month, okay, through the use of these interlocking blocks. So that's a nice area that's going on. We also do all of this in silico. So everything that we saw in the scaffold design, that is the finite element model, and going back from it, the mesh comes from the CT scan, and the properties of the mesh come from molecular dynamics. The degradation of scaffold is captured using some degradation models that are empirical models. So this whole thing has a predictive capability. Essentially, we want to be able to make metastasis predictable. I have a friend who 3 years ago I had lunch with her, and she had breast cancer metastasis, and she told me she thinks she has about 3 years to live. That was in May, and in July I attended a funeral, so she had no idea. That's how unpredictable it is. So we want to change that, and we are making some progress towards it. This is a finite element. So I said a lot of different things. Basically what we have made is a bone mimetic in vitro model, a test bed of prostate and breast cancer metastasis. The test bed is actually tuning the bone microenvironments. It's influencing the bone through mechanical and biochemical cues. We are also using a bioreactor and using the application of shear forces. And this test bed can be used to discover biomarkers of cancer metastasis. I showed you 2 examples using IR and Raman, and also mechanobiology-based, mechanics-based. And it's an important problem to continue to pursue, because 1 million people die due to breast cancer and prostate cancer worldwide each year. And I particularly want to say that it's really important, given the different tools of materials characterization we have, and the tremendous modeling and computational capabilities that we have today. It's really important that engineers make efforts in this area. It's really important. Now, engineers always have made the devices of medicine, but I think in this problem, it's very, very important to take more engineering approaches towards this. And these are, of course, all the people that did all the work, and I just get to travel to fun places like Miami to come and talk about it. So, thank you.
Thank you very much. It's a amazing story. I would say research story, starting with the building the bones to actually applying it. So yeah, no questions. I think very good presentation.
Thank you for that title. It was amazing. But biomedical engineering is kind of drew me. You did so many different things at so many different levels from a very engineering standpoint. So thank you for that. My question was about your scaffold and the tumors or mesenchymal stem cells adhesion to it. You talked about how your scaffold is made of this very heterogeneous structure of the nanoparticles within the main structure, and then the hydroxyapatite nodules. And then you're trying to model the adhesion of these actins, these integrants, to either the clay or the hydroxyapatite.
Yes.
In your studies, do you notice a difference between the cells wanting to attach more to a part of your heterogeneous structure, or is it homogeneous enough where the cells can't tell the difference of what they're attaching to?
Yes, so that is not published yet. So right now we have some data that's completed on silicates. So certainly silicates have a role. I will only say that much right now. Certainly silicates have a role. So does hydroxyapatite. Keep in mind that we have our scaffold is basically degrading, so the polymer is no longer relevant at this point. So it's replaced, so that's why we waited 23 days before applying the cancer. So we really don't have any very much of the scaffold remaining. It's just bone cells and tissue. So bone is in itself heterogeneous, right? So if your question is collagen versus the mineral, right? In the bone,
Sure.
definitely mineral. Definitely the mineral is playing a role. The reason we are looking at the silicates is because we're looking at it from a therapeutic point of view. That what can we do to influence the integrities? I can only say that much right now. But stay tuned.
It was really cool work. Thank you. So when you're thinking through, when you're flowing your cells through, and you're looking at the ones that say adhere versus don't, is that completely stochastic? Are you comparing ones that do adhere and integrate with the bone scaffold versus ones that don't? Or is there something fundamentally different between those two cells?
So everything in any time we work on cells, everything is statistics. So we measure how many have, so of course, even when I say MET, I don't mean every single cell. It's statistically higher number that have undergone. So same goes for adhesion. Again, that falls into an area that's ongoing, and I didn't show. We have a very unique instrumentation with we are measuring adhesion forces. Then we would be able to distinguish between the cells that, let me just repeat that again. We are able to, so just basically an AFM to measure the adhesions. We have, our unique system has a confocal attached to it. So we'll be able to look inside the cell to where the actins are. And so that is still ongoing. So what happens to the cell that adheres compared to the cell that doesn't adhere? How activated is the integrin? We don't have the answer to that yet, but everything I showed is statistics.
Have you looked into accounting for stromal cells, particularly fibroblasts, macrophages, other cells
Yeah,
like that?
thank you. Thank you. I always get asked that. So as we are making the test bed more and more realistic, one of the things missing in that is very aptly pointed out is the type of cells. So here we have just shown MSCs, mesenchymal stem cells. Obviously there's osteoblasts, and then there's the cancer cells. But in the real life environment, there's many kinds of cells. So there is also ongoing. So right now we have some data using endothelial cells. So I just want to say we are duplicating both the primary site and the secondary site, and we're causing the flow of the cells. So yes, that is ongoing. I didn't show any data.
Certainly the fibroblasts will contribute
Yes,
to the extracellular matrix.
Yes.
And the extracellular matrix is going to have a big contribution of everything else that's going on in the environment.
Yeah, that was also a comment on the last proposal submission, and that is actually ongoing work on our NSF Center research. So I have some collaborators who are cell biologists, so cancer biologists, and they have already developed a co-culture. Now we're trying to see how to put that, the best way to put it on the scaffold. And that has a fibroblast, so that's 3 other types of cells. You're absolutely right. We want to get as close as possible to real life. Thank you.
Any other questions from the students?
So I do have another question. Are you able to get biopsies and patient-derived tumors from metastases, from bone metastases? Because that must be pretty tough.
Yes, thank you for asking. I am so happy to. Why, that sounds really mean. Because somebody had, we have not been able to get for breast cancer, but we have, as of today, we have 3, and I mean literally as of today, because the third one I heard this morning, we have 3 viable cell lines of bone metastasis prostate cancer. One of my collaborators, Dr. Vyas, is the orthopedic oncologist, the only one in South Dakota and North Dakota. He's like the primary, and he was personally involved with this project, and he was able to get us 3 cells. So we have right now 3 cell lines of both
So 3
bone.
patient-derived that
Patient-derived.
that have
We
been
haven't put
converted
those
to
on
lungs.
scaffold yet.
No, but they've been converted to lungs, they've
Yes,
converted to.
yes, both for bone and for, but you are so right, it is so difficult to get. So first, the patient has to, not only they have to undergo the surgery, so my students are waiting with the media and everything set up, and then I get a phone call and they go over, it's only 20 minutes away, in the Fargo traffic. But they go over to the hospital and then they're told whether the patient consented. If the patient consented, then they arrive there, but then pathology takes the tissue, we only get the leftovers, so patients are not undergoing.
Because bone metastases must be very, very difficult to
Yes.
obtain, because they're not typically, unless you're gonna remove part of the bone, which happens.
We have bone also, so we are saving that, we have some bone chunks. The surgeon explained how he does that, is there's a little auger, and then he goes in with a tube, and he takes all of the material out, and that is the tissue, the soft tissue, and the bone, and then we have to separate it. Unfortunately, in the process, he's going through healthy bone, so that has also got fragments from that. So we have this very complex, we were able to isolate at least the cancer cells from it and then turn them into cell lines. We have 3 as of today. So it is extremely difficult to get bone met samples.
Will you be using the horizontal bioreactor when simulating the cell?
Yes, we will. We are right now, the experiments going on in the lab are with the commercial cell lines. The patient-derived cell lines are much too precious. So we're perfecting the process with commercial cell lines. So that work is soon to be completed.
Okay, let's thank the speaker for an amazing talk.